So I probably switch a little. I do not do the simulation. In this talk, I basically just report the experiments we recently did for tumor cell adhesion, but relate to the endothelium structure. So first, I would like to show you what is the endothelium surface glycocalyx. On the endothelium cell, which form every of our vascular wall, is covered by a layer. You can think about a brush like layer or fluffy layer, something just from the appearance. This is an image from electron microscopy view from Squaranto in 2001. This is surface glycocalyx on the post-capillary venule of rat mesentery. And from this scale, 150 nanometer, you'll guess roughly the thickness of this layer is 100 nanometer. But this is from electron microscopy view. So for preparation for electron microscopy, you have to do a lot of preparation, dehydration, right? So that's roughly the thickness from electron microscopy. So here I just give you a cartoon. This endothelium cell lining the vessel wall. This is tissue space. This is blood. So because of its unique location, the surface glycocalyx, it can play several roles. It can serve as a mechanosensor in the transducer. Also, it can maintain and modulate the microvessel wall or vessel wall permeability to water and solids. And the third one, because it's a barrier between interactions of circulating cells like leukocytes, platelets, tumor cell, and endothelium. So because of these important roles, a lot of people like to investigate it. So for the pathologic stage, people find this surface layer was damaged during many vascular diseases, including atherosclerosis, diabetes, and other cardiovascular diseases. So since 1966, people started to just wanted to know the thickness and the organization of this layer by different methods and in different type of like vessels in capillary, like aorta, arteries, in different species, like from mouse to human. They found the distance or the thickness of this layer ranges from, like this one is 20 nanometer up to 8.9 micron. This is all the real vessel. And of course, they use different methods and different fixation methods and the visualization methods. They use transmission electron microscopy, micro-PIV, intravital microscopy, Confocal, and the multiphoton, and some other methods. So, and in our current study, we want to know why there are a huge difference. This is 20 nanometer. This is 8.9 micron, 1,000 times difference. So even though we understand because of different preparation and different visualization method will induce this kind of discrepancy, then can we do kind of like a better method, especially for like a fresh or live vessel? So that's the initiation of the first project. So here I show you a cartoon for molecular composition of this surface glycocalyx. This is the lipid layer of endothelium cell. We can see there are a lot of proteoglycan and glycosaminoglycan in this layer. And in this one, I give you some kind of example. This blue dot line indicates heparin sulfates. And this yellow one is hyaluronic acid. And this green one is conjugated sulfates. They're all GAGs, glycosaminoglycans. And most abundant one is heparin sulfate, but of course we do not show the percentage of each component. So in our case, we want to see if we use antibody with the furansen to attach this heparin sulfate, can we at least tell the thickness of this layer in vivo? Okay, now this is our animal study. So we use the microvessel on rat mesentery or the mouse cremus muscle. Rat mesentery is very easy preparation. We do not even damage anything. But for cremus muscle, we probably have to do a little surgery. And for our preparation, we keep the animal alive. We use our, we call it micropipette with the 2 lumens, theta pipette. And the tip of the theta pipette roughly 30 micron or less. Of this one only like 20 micron. And then 1 lumen of a pipette we have filled with the blocking solution or washout solution. And the other side we filled with the furansen labeled like anti-heparin sulfate. So this theta pipette is collected to a water manometer. So in this way, if we increase the pressure on the blocking or washing side, we can perfuse solution into arterial, several, into an artery collecting to several arterioles, capillaries, and post-capillary venules. And if we switch to the labeling side, we can perfuse antibody. So roughly, we cannulate the artery size probably 25 to 35 micron. So we first blocking, and then we inject or perfuse this FITC anti-HS into the group of microvessels. Under this condition, superfusate, we have another dripper to supply the superfusate for 2.5 hours under 4 degrees Celsius, because we try the body temperature, we try room temperature, won't stay until we reduce to 4 degrees Celsius. Probably that's the optimal temperature for those kind of antibody like conjugation. So this is our technique. So after that, we observe the surface glycocalyx labeled with the anti-HS by confocal microscopy. For this is one cross section. And then for one cross section, we have 8 locations. We get it. And then if I draw a line, the red line, I get 2 spikes. And we define the thickness of this layer is the distance between 2 points which have the half maximum of the highest intensity. That's the thickness. For this case, roughly 1 micron. So here's a summary for rat mesenteric microvessels. So this is one capillary. This is a mid-plane view. This 3-dimensional view, this cross-sectional view. And we have arterial, capillary, post-capillary venule. Surprisingly, we do not see the anti-HS in arterial. And then this is the size of these arterioles. But for capillary and the post-capillary venule, we do see like about 1 micron thickness surface glycocalyx. And the same is true for mouse cremus muscle microvessels. We still do not see the anti-HS in arterioles, but we see significant, roughly 1.5 micron in the mouse cremus muscle capillary and post-capillary venules. So that's the in vivo immunostaining for surface glycocalyx. And we also wanted to know if, like in the length scale, like 100 micron, we want to see if the glycocalyx distribution is uniform or not. So we have like this one segment 120 micron, and then we do an integration, find the intensity. So you see the green one in the dark region. So we have a normalized intensity. If this segment, like with N, if the average normalized intensity in this segment is less than 0.2, we define the surface glycocalyx is like negligible. If it's about 0.5, it's Y segment, then we see you have significant surface glycocalyx. So if we use this criterion, we find roughly for post-capillary and capillary in rat mesentery and mouse cremus muscle, we find half of the vessel segment are covered with the surface glycocalyx in half or not. And roughly like every like 30 to 50 micron, you see like a missing part. So here is a summary for the first part. We developed a new method to directly quantify the surface glycocalyx thickness in microvessel, that's in vivo. And we found that there is like about 1 micron thick surface glycocalyx in rat mesenteric capillary and post-capillary venule. And there's 1.5 micron in mouse cremus muscle capillary and post-capillary venule. However, we do not see the anti-HS in arterioles in both mouse and the rat. And we also found the surface glycocalyx is not uniformly distributed on the microvessel wall. So that's the first part of my talk. You see there's significant amount of surface glycocalyx in the capillary and the post-capillary venule of rat mesentery and mouse cremus muscle. Okay, now this is my second part you're interested in. The tumor metastasis. I think probably everybody in this room knows the cascade of tumor metastasis. Here I just summarize. First, the primary tumor need to get into the blood circulation, called intravasation. And then they go with the circulation blood, so they call it circulation tumor cell. They adhere, rest somewhere in a distant organ. So this is a lot of story. Seed and soil, or the flow pattern, or geometry, or many things. Okay. And then when they adhere, tumor cell adhere to the microvessel wall, usually they get out from the microvessel, especially post-capillary venule. They hardly get out from arterial. Okay. And then if they have interaction with the microvessel wall, they should probably induce certain damage or something. We have to assume. And then finally, they get out. Because this surface glycocalyx, that's blue layered, is the first barrier for tumor cell to overcome to go extravasation. That means they have to do some kind of degradation. So here's surface glycocalyx. This is tumor cell. They have to go over this layer. So based on this kind of thinking, we propose the hypothesis. We hypothesize that tumor cell adhesion can degrade the endothelial surface glycocalyx, which can increase microvessel permeability to water and the solids, and can also further increase tumor cell adhesion. So to test these two hypotheses, we have these aims. So first, we have to determine initiation time, how quick this type of like MDMB-231 is a very malignant human breast cancer cells. So we want to use this one to say is how quick it can adhere to the microvessel wall and the accumulation rate, adhesion rate in post-capillary venule. And 1 millimeter per second, that's the mean blood flow velocity in this type of vessel. We also do the calibration. So first, we understand the tumor adhesion and adhesion rate. Second, we have to quantify the microvessel permeability to water and the solid. We pick up albumin. That's pretty large molecule, which usually cannot pass the microvessel wall. And we also wanted to measure reflection coefficient to this solid, because reflection coefficient, that's the parameter which indicates the selectivity of the microvessel wall. So that's all transport parameters. So we measure this permeability and the initiation time of tumor cell adhesion and after 45-minute perfusion. Of course, we also measure under control condition without tumor cell adhesion. And then we use, this is one we have a little mathematical modeling for the gap between endothelium cells. Then we predict most likely structural changes based on our measured permeability data. And after that, we want to confirm our model predictions. Then we quantify the glycocalyx and microvessels under control in the initial tumor cell adhesion and after 45-minute perfusion using the same method as I just showed you. Okay, now we wanted to do the tumor cell adhesion experiment. We pick up one post-capillary venule, roughly like 35 to 50 micron in diameter. We still use the micropipette. But inside, this is from mesentery. Inside micropipette, we have our tumor cell. It's still collect to a water manometer. And we control the velocity. And then we inject tumor cell with this post-capillary venule. And then record its adhesion. Here I just show you a little movie. Then, of course, we have to label the tumor cell with the fluorescent dye, otherwise we cannot see it. So then you see this is the perfusion tumor cell, and then you see the accumulation of tumor cell on the microvessel wall as I turn on the bright field. So you see it. So this one is not real tight. It condenses 1 hour adhesion into this like 1 minute or less movie. So this is our experiment. And here is a summary for 14 experiments. So roughly, like in 7 minutes, we see one adherent tumor cell in about like 100 micron non-vessel segment. And then we do a 1 hour cell adhesion. So roughly in an hour, there are 7 adherent tumor cells in 100 micron non-vessel segment. So pretty linear. So this is tumor cell initiation time and accumulation rate if we find the slope. So after that, we have to measure the permeability. Here I show you how do we measure permeability. It's a classic 90s technique. So this is a microvessel that we use post-capillary venule. This is a micropipette with a marker cell. We use red blood cell as a marker cell. So we collect this micropipette to a water manometer, and then we know the perfusion pressure. So if downstream of this microvessel is occluded, so if the vessel wall is impermeable to water, and then you do not see the movement of marker cell, and you probably see the expansion of the microvessel. However, if the vessel wall is permeable to water, and then the water is out, it's the flux, and then you'll see the slow movement of this marker cell from the cannulation side to the downstream. So from this movement, speed, and the length between cannulation side to the occlusion side and the radius of the microvessel, we know the flux of water out. And we know the water out is also determined by the driving force, the hydrostatic pressure difference and the osmotic pressure difference across the vessel wall. And the coefficient we call hydraulic conductivity. That's microvessel wall permeability to water. So if we plot, we change the driving force delta P, and we record the marker cell movement speed, then we can get this flux versus driving force line, and the slope is LP, and the intercept, that's the reflection coefficient for albumin, because inside the perfusate, we also put the certain concentration of albumin, so we know this delta pi. And we can calculate reflection coefficient to our solute. So here I just show you the real experiment. This is the micropipette, this is the vessel. Inside, you have a microcell. So now we perfuse if we increase the pressure. And then the downstream is open. Okay, if downstream is occluded, we still see the slow movement of microcell. So from this movement, we can determine the LP and the sigma. Okay, and then we unblock it. So we find this tumor cell adhesion can increase hydraulic conductivity LP. So here I show you, this is the normal one without tumor cell adhesion. This is when tumor cell just adhere, initial adhesion, just one tumor cell, 100 micron microvessel segment. So, and the hydraulic conductivity increased by 1.6-fold compared to the control. So after 45-minute perfusion, we remeasure. The LP increased by 2.7-fold. Okay, this is for LP. And how about diffusion? How about reflection coefficient? We find the control, the selectivity or reflection coefficient to albumin is 0.92. And the initial tumor cell adhesion decreased 0.64. And after 45-minute diffusion, decreased further to 0.42. Okay, this is the experiment for LP and the sigma. And then we have another one. So now you ask me why I want to do a lot of measurement for permeability. I will tell you later, because different type of permeability will tell you the change in different structural components in the microvessel. That's why we have to get more. This one is we measure solid permeability, microvessel wall permeability to a solid. So here is the vessel. On top of the vessel, we have a measuring window. So still use theta pipette, and then we inject the dye, and we see the step increase of the fluorescent intensity in this measuring window. A step increase. If the microvessel wall is permeable to this solute, and we see the accumulation of the solute in the tissue and increase the total intensity in this measuring window. And then from this step increase and this slope increase, slope of this increased curve and the radius of this vessel, we can calculate solid permeability. Okay, then I show you another movie. So this is a measuring window, and then if we inject the dye, so you see it, but you cannot see by your eyes the diffusion of the solid into the tissue space. Okay, wash out, and we can repeat experiment. Okay, this is for solid permeability. This is for albumin permeability. So this is control initial adhesion increased by 2.2%, 2.2-fold, and after 45-minute perfusion, we got 5.6-fold increase. So why I want to have this data? Okay, later we have to check if, because we perfuse tumor cell for roughly 1 hour, and we see if the secretion by tumor cell will increase the permeability. Then we get the supernatant from the 1 hour this type of tumor cell. We measure permeability. It doesn't increase significantly. Okay, so here I gave you the summary for the experiments. And first we said initiation time for this type of tumor cell in post-capillary venue is about 7 minutes, and then it's linear increase, and the rate accumulation or adhesion rate is 1.2 cells per 100 micron vessel segment in 10 minutes under the mean blood flow velocity in this type of vessel. And then the initiation of tumor cell adhesion, we see the increase for LP, reflection coefficient, the solid permeability, and after 45 minutes, increase even more. Okay, but this is lumbar, we are going to use it. So that's the reason I labeled here. But the super latent for incubating this tumor cell for 1 hour does not increase the solid permeability. Okay, now I show you the part by mathematical modeling. So for water and hydrophilic solids, people find 90% of them will go across the microvessel wall through the interendothelial cleft. That's a gap between one endothelium cell and another one. So this is surface glycocalyx. And inside you have junction strand with the pores and ultrastructure data from NIMBioS in 2004 for the same type of post-capillary venue. Okay, this is a model geometry. Based on model geometry, we can calculate the permeability to water in the solids. Okay, and here is our prediction. So the first one, so it's if we remove surface glycocalyx, and the LP will increase only by 2 point something. And then, but the albumin permeability increase much more. Is this trend if we think tumor cell adhesion degrades surface glycocalyx? Okay, that's the first case. So now second case, you're saying probably the tumor cell adhesion will increase the gap width of the interendothelial cleft, or increase the pore size, or increase number of pores, right? Okay, now we have the prediction. So this is why if we increase the gap width of interendothelial cleft, LP increase much more than albumin permeability. And then if we increase the pore size, then LP still increase much more than albumin permeability. Increase the number of pores, still LP increase much more than albumin permeability. So compare to our measured data, we find this case is most likely case. That's the reason I put this number. It's initial adhesion, the LP increased by 1 point something fold, and the albumin permeability increased 2.2 folds. So this correlates to roughly, you only have like 44% of surface glycocalyx left, like roughly 60% will be removed. After 45 minutes, tumor cell perfusion, then you see the LP increased by 2.7 and the albumin permeability increased by 5.6, and this correlates roughly, you only have 10% left, so roughly 90% will be degraded. So this is our model prediction. So, and then we have to confirm if the surface glycocalyx is really degraded by tumor cell adhesion. So we still label the HS. So, and then we use the same method as I just showed you, but under control and the initial adhesion and the perfusion for 45 minutes. And here is our results. Under control, you see a lot of green. That's surface glycocalyx. And then this red is adherent tumor cell. So the initial adhesion, you see less green. And then after 45 minutes, tumor cell perfusion, you see more red, more adherent tumor cell, but much less green compared to the control. So if we give you a little summary, so then initial adhesion will degrade surface glycocalyx to 44% of the control. In the 45-minute, tumor cell perfusion will degrade to like 18%. So in this way, we'll think about, can we preserve this surface glycocalyx by certain type of aging? Now we use the orthomycoid. That's the natural plasma protein. So we pre-treat it, and then we'll say surface glycocalyx increased by 40%, and it can reduce the solid permeability. It's really increase the barrier and also reduce the tumor cell adhesion. So that's the one method. We preserve our microvessel wall surface glycocalyx. In this way, we can prevent tumor cell adhesion. So at least we self-protection. So this is our conclusion. Our model predicts that the increase in LP in solid permeability and decrease in refraction coefficient to solid by tumor cell adhesion is most likely due to the 60% degradation of surface glycocalyx in the initial adhesion and the 90% degradation of surface glycocalyx after 45-minute perfusion. And we use immunostaining with FIGHT44. See, this was very much match each other. So still that 44 and left 18%. So the agreement between experimental results and model prediction suggests this type of tumor cell adhesion increase microvessel permeability by degrading the surface glycocalyx in the microvessel wall. If we preserve this surface glycocalyx, we can reduce the solid permeability as well as tumor cell adhesion. So here I'd like to thank my student Wang Yiyan, who did the first project, Bin Cai, and Jeff Fan did the second project, and our lab technician. I also like to thank our collaborator, John Tavier, and the City College in the first project, and Dr. Filippo Gilcati, and the Memorial Sloan Kettering Cancer Center for the second project. And I'd like to thank the funding support from NIH and NSF. Thank you very much for your attention.